Well, thank you very much for that kind introduction, and it's an honor to be here today. I've been asked to talk about the NIH perspective on personalized nutrition and precision medicine, and that's a lot to cover in 10 minutes. So I'm really going to give a lot more of an overview, give you some of the websites and what are some of the things going on, and then you can go and find more information if you're interested. So I have nothing to disclose, but I should say what I'm about to present are my thoughts and opinions and not necessarily those of NIH. Also, I do work for NIH. So the genetic revolution is providing the tools to understand the effect of a lot of different things, understand the relationship between nutrition and disease prevention, and understanding genetic variants provides the tools to understand differences in disease susceptibility, as well as differences in response to dietary components. Emerging genomic technologies are really providing the tools that we can have a shift from making public recommendations to more personalized recommendations. So what is personalized medicine? It is using information about a person's genetic makeup to tailor strategies for the detection, treatment, or prevention of disease. Examples relevant to the nutritional community are infant screening for PKU. Other examples include screening for mutations in BRCA1, which is associated with an increased risk of breast and ovarian cancer. It is using molecular profiling technologies to assess DNA, RNA, protein, and metabolites to tailor medical care. And really, this approach has the promise of delivering the right dose for the right indication to the right patients at the right time. The problem is, with many different diseases, we really don't know how to prevent or treat them. And so NIH is moving into the era of precision medicine to hopefully to start developing some of the tools. So what do we mean when we talk about precision or personalized medicine? Well, it is a groundbreaking approach to disease prevention and treatment based on people's individual differences in environment, genes, and lifestyle. And the hallmark of this initiative is the All of Us research cohort. And it will try and lay the foundation for using this approach in clinical practice. This cohort hopes to engage 1 million or more US research participants who will share biological samples, genetic data, and diet lifestyle information, all linked to their electronic health records. And this data will allow researchers to develop more precise treatments for many diseases and conditions. The research is based on the cohort data, will lay the scientific foundation for precision medicine, help identify new ways to treat and prevent disease, and test whether mobile devices such as phones and tablets can encourage healthy behaviors. And the whole goal, as I said, is to develop the right drug for the right person at the right time. So why are they doing precision medicine now? What was the push? Well, the NIH felt that the time is right because we have greater understanding of the human genes. People are more encouraged in healthcare and research. We have the tools to track health information and use large databases, and research technologies have improved. On the bottom of this is a website, and if you'd like more information about this initiative, as well as if you want to be a participant, it can show you how to sign up for it. Similarly to the Precision Medicine Initiative, there's also a large interest at NIH on environmental influences on child health outcomes, or the ECHO. And ECHO is pretty similar, except their cohort is actually having multiple existing cohorts that are actually working together to try and look at the environmental factors on child health and development. The environmental factors include physical, chemical, biological, behavioral, and social determinants. The ECHO program will allow researchers to maximize the use of existing resources, leverage available data sets to develop a repository, and use this information to predict disease development and test new tools and approaches. In addition to the information that the individuals are collecting as part of the regular cohort, there are certain standardized information that they'll be asked to collect. And these include information on demographics, health, typical health and development, genetic influences on child health and development, environmental factors, and patient-reported outcomes. And the 4 main diseases that they're initially looking at are upper and lower airway, obesity, pre, peri, and postnatal outcomes, and neural development. And again, more information about this can be found on the website on the slide. So now we move into the question of precision or personalized nutrition. And can you use the same tools? And can your genes tell you how to focus your diet for disease prevention? And really, to phrase this another way, is it logical to assume that everybody will respond the same to dietary components for disease prevention? Or will differences in your genes influence how you respond? Well, nutrigenomics is a discipline that investigates the effects of dietary components on the structure, function, and regulation of coding and non-coding DNA segments of all genes present in the genome of a given species. And it has been endorsed by the food industry, as shown by this cover of Food Technology. In fact, I made this slide with John Milner about 10 years ago, when we really believed that if we started understanding nutrigenomics, that would actually provide the tools to start identifying responders and non-responders to diet for disease prevention. Nutrigenomics encompasses an understanding about how genetic variation influences the response to dietary components, which can be studied with nutrigenetics. Nutrient-induced changes in DNA methylation or in chromatin alterations such as histone acetylation, which can be studied with nutritional epigenetics. How dietary components change the rate of transcription of various genes to RNA, which can be studied with nutritional transcriptomics. All the proteins present in a particular cell at a particular time, which can be studied with proteomics, and all the metabolites at a particular time, which can be studied with metabolomics. Well, what we've realized is that these tools are not enough, and they do not provide all the tools that we need to start looking at personalized nutrition. And I'd like to say that I believe that metagenomics might actually be the missing piece of the puzzle. And in fact, we've realized that a dynamic relationship exists between the gastrointestinal microbiota, our response to dietary components, and the intestinal epithelial cells. And in fact, both the types and numbers of microbes, as well as the specific dietary components that we eat in energy, can influence specific microbial metabolites, which can have a role in disease risk. And in fact, metagenomics, which is the study of both the genomes of the microbes and of their mammalian host, might actually provide the tools to start being this missing link in the puzzle of understanding personalized nutrition. And in fact, a landmark paper published earlier this year from Israel highlights that this might be the case. In this study, they looked at the personal glycemic response to individual foods in a cohort of 800 individuals. And what they observed was that the individual response really varied among individuals. You could give one person a banana, and they'd have a low glycemic response, but a very high glycemic response to a cookie. And you could give another person the cookie, and they'd have a low glycemic response, but a very high glycemic response to a banana. So what they did was take typical blood measurements, i.e. their glycemic response to different foods, anthropometric data, food dietary information, and analyzed their microbiota, put it in the computer, developed an algorithm, and then validated this in a cohort of an additional 100 people. Moreover, they found that it was actually better than traditional recommendations in understanding the glycemic response to different foods. They did a validation study, or a controlled feeding study in 23 individuals, and they actually found that this approach worked. So the question is, can we utilize this type of approach of integrating information about our microbiome with our dietary intakes and developing algorithms to better understand susceptibility to other diseases? Well, this interest in a personalized approach actually expands beyond NIH. The federal government released on March 4th, 2016, the first 5-year roadmap, and it encouraged an increased focus on research that can lead to more individualized advice for promoting health and preventing disease. The roadmap identified 3 key questions that cover a broad spectrum of research, and the 3 questions were: how do we better understand and define eating patterns to improve and sustain health? What can be done to help people choose healthy eating patterns? And how can we develop and engage innovative methods and systems to accelerate discoveries in human nutrition? And out of these There were a number of different agencies involved, as you can see on the top. These 3 questions were divided into 11 sub-questions. And question 1, topic 2, the individual differences, including Omics, really gets at this whole idea of personalized nutrition. And there were 5 agencies that were interested in this question. NIH is trying to utilize this information from the roadmap to try and say, how do we develop a strategic plan for nutrition research? And in October 2016, they started the Nutrition Research Task Force. Chris Lynch, the director of NIDK's Office of Nutrition Research, will serve as the executive secretary. And the goal of this group is to guide the development of the first NIH-wide 10-year strategic plan for nutrition research. The task force is chaired by the directors of 4 NIH institutes, and they will have a senior leadership group to guide implementation once the plan is complete. This slide shows the timeline for the planning for NRTF. As I said, we're just in the process of getting scientist input on it, but the one nice thing about this new NIH roadmap for nutrition research is we will try and get input from all interested stakeholders throughout the process, and we expect it to be released in October of 2018. ODS also recently underwent a strategic planning process, and their strategic plan is on this website. Well, I'm out of time, so just to quickly summarize, in 2008, Ilya Serhouni, when he was NIH director, in a statement to Congress, really talked about the future being personalization, and he talked about the 4 Ps. And it's important that any future research be predictive, personalized, preemptive, and participatory in the sense that all people are involved. And I really believe the recent NIH initiatives, such as precision medicine and ECHO, are really start to move in this direction. Thank you.
Thank you.